So the moment we've all been waiting for, finally, section 7, mitosis. So we've been talking about when you do mitosis, why you do mitosis, how bacteria do binary fission instead of mitosis. Anyway, now we're finally talking about mitosis, which is the division of the nucleus, also called karyokinesis, taking the nucleus and breaking it, dividing it. So we're separating out your nucleus. So every time you need to make new cells in your skin, for example, you're going to do this. So this is cell division in eukaryotes. It's always asexual, meaning for what I'm talking about here, meaning you're not making egg and sperm. You're just making new skin or liver or bone cells or whatever. And so here are the various stages. You've got prophase here, and what's happening is that the chromosomes are condensing. And this is like a movie, and I've stopped and just taken snapshots. So the stages aren't like they have a stage and sit around. The stages of mitosis are continuous with each other. So the chromosomes condense, then they meet here in the middle, and then they're pulled apart. And then you get two nuclei at the end. So the purple, or I guess it's purple stuff here, are chromosomes, and the red stuff are the spindle fibers. They're part of the cytoskeleton of your cells, pulling apart the chromosomes in this case. The video will never work in this. Okay, mitosis. So here are the stages of mitosis, and this is the way it's written in your notes, so I want you to label them. This is called prophase, and this is called metaphase. And so they go like this. This is the first one, this is the second one, and then over here is the third one. They're being pulled apart, so that's anaphase. And here's the fourth one, which is telophase, or telophase. And so prophase is when they're starting to condense. Metaphase is when the chromosomes are in the middle. And so that's nice because middle and meta start with the same thing. Anaphase, they're being pulled apart. And they both start with A's. And then telophase, I don't know, sometimes I still tell students it's the, and I don't know if that helps you or not. It's not quite as good as middle metaphase and ana apart. So mitosis is division of the nucleus, and it's also called karyokinesis. Again, a prokaryote, a eukaryote. So for example, let's look at this word, eukaryote. Eu means true, like well, true, and then true nucleus, like you or a plant or a fungus or a protist. Anyway, karyote is nucleus. So karyokinesis means the nuclear division. You're separating out the nucleus. When we're done with this, we'll talk about cytokinesis, and this is a division of the cytoplasm. This doesn't have to be perfect. Karyokinesis has to be perfect. You've got to get exactly the same amount of DNA to one side as you do to the other. Each new cell is going to have a complete perfect copy of the DNA. Anyway, this only occurs in eukaryotes. If you're in a prokaryote, or if you are a prokaryote, a bacterium, prokaryote, you're going to do something called binary fission because you are simpler and you don't have the same kind of DNA. Okay, back to mitosis. There are 4 stages. This doesn't occur in some mature cells like brain cells. They go into something called the G0 phase, which I'll show you at the end of the unit. Anyway, so the 4 mitotic stages, we've got prophase, and the next stage, which kind of happens continuously with the first one, is metaphase. Then you roll into anaphase, and then you roll into telophase. I have some videos of this that I'll show you at the end, but you can't see it in my screen capture. So if you just Google mitosis videos, you'll come up with a ton of stuff. Anyway, the order is PMAT, prophase, metaphase, anaphase, telophase. So I've memorized PMAT, and so should you. You can also memorize IPMAT, I for interphase, but only this part is mitosis. The I part is interphase, which has the gap 1, the synthesis, and the gap 2, and that's not part of mitosis. Overview of mitosis. Again, prophase. In metaphase, they line up in the middle, right here. In anaphase, you separate out your DNA. That's the next stage. And telophase is when you have 2 new nuclei at the end. And this one's not actually mitosis. Oh man, that's cytokinesis, where you're separating out your chromosomes. So these are mitosis, and this is cytokinesis, which is the stage after mitosis. So some definitions to learn. So here's the planet, and let's see, this is a lousy picture of North America. Oh man, oh, that's an awful picture. Aren't you glad I'm not like a geography teacher or something? Let's see. All right, that's the planet. Oh man, that is awful. Anyway, you've got poles, the North Pole and the South Pole, and then you've got the equator, which is probably drawn in the wrong place. But anyway, the equator is the center of the cell, and it's just like the center of the planet. So the equator is the center of the nucleus. I shouldn't have said cell. Center of the nucleus, where the chromosomes line up. And then, yeah, let's get that out of the way. Here it is. And then you also have poles at either end. So the poles, whoa, really? So the poles are places where centrioles go during mitosis. Each will become a new daughter cell. So it's um, the opposite ends of the nucleus, which is just disintegrating by then. Anyway, okay, so now we're going to go over each of the stages of metaphase. Remember PMAT. And so we'll talk first about P for prophase. Prophase is kind of a long stage, so sometimes we separate it out into early prophase and late prophase, so that's what I'm going to do here, but it's all prophase. So the chromatin in the nucleus condenses to form visible chromosomes. The reason you've got to do that is if you've got a cell, and here's your nucleus, and here's your DNA, and it's all spread out, if you're going to take this DNA and spread it out that way, by the time you get this part of the DNA over here, the other rest of the DNA will still be over here. You've got to cleanly separate them out so that you're not breaking it, and when you pull a chromosome over, you're pulling the whole thing over at once. So that's why we don't do it like that. That's why we make these chromosomes. We condense them so that they're easy to separate out. So you form visible chromosomes. The mitotic spindle forms from fibers in the cytoskeleton. And they form from, should probably say, from or organized from centrioles in an animal cell. Plant cells have a different way to organize their cytoskeleton, but for us, you can learn about centrioles. Remember the cytoskeleton from our cell unit that has a bunch of microtubules, microfilaments, all that stuff. It's the proteins that hold your cell to have the right shape, so that if your red blood cell is shaped more like a snow tube instead of more like a circle, like a sphere, for example. It also helps move organelles around. In this case, the part of the cytoskeleton, the spindle fibers, are used to pull chromosomes apart. So spindle fibers are a part of the cytoskeleton of a cell. And so here's a picture of early prophase. You've still got your nucleolus, the cell membrane, the nuclear membrane, sorry, here's the cell membrane out here. The nuclear membrane is going to start to disintegrate pretty soon. But you can see the chromosomes just starting to condense. So there's the nucleolus, it'll get broken down soon. There's the cytoplasm, all the goop. Your cell membrane, and this is a plant, so cell wall, are out here. And the nuclear membrane's still there, but it's about to disintegrate. So late prophase, the nuclear membrane and the nucleolus are broken down. Remember, the nucleolus is the little structure in the nucleus where, let's draw a picture. Here's the nucleus. Here's the nucleolus. So it's a little structure that makes ribosomal RNA to be brought out to make ribosomes. But if your DNA is all condensed, you're not making RNA, and that RNA isn't going to be making, that RNA wouldn't be making this kind of RNA either, right? That RNA wouldn't be ribosomal RNA. So help yourself, kiddo. And so here you can see the nucleolus is gone, and you can see condensed chromosomes. Chromosomes continue condensing and are clearly visible in late prophase. The centrioles migrate to the poles, so that's at least in an animal cell. Here's a centriole and another centriole, and look, they've migrated to the poles, and they're starting to connect to the chromosomes, the kinetochores of the chromosomes. They're not lined up in the middle yet, but they're getting there. So the spindle fibers attach to the kinetochore of each chromosome. Here they are. Here's the kinetochore, and here's the spindle fiber, and this is the centriole. And then the spindle fibers finish forming between the poles of the cells. So you actually have spindle fibers here which help to separate out the poles from each other. And here's a picture up close. So spindle fibers attached to the chromosome right at the kinetochore, or also called, the region is called a centromere. So here's the spindle fiber, again, part of the Cytoskeleton. So cytoskeleton fibers, the more specific name in this case now is going to be spindle fibers. So there's lots of cytoskeleton fibers that do other stuff, like hold the cell in to have the right shape. Spindle fibers are part of the cytoskeleton that specifically pull chromosomes apart. So there are the spindle fibers, which have formed, and they form from microtubules. So there are various proteins that they form from. And then the next stage is metaphase. Yay, we've made it to metaphase. And metaphase is a quick stage because really all that's going on is the chromosomes have lined up in the middle. So the chromosomes attached to the spindle fibers move to the equator, or the middle of the cell. Here are the poles. If it's not labeled in your notes, you need to label poles here and label equator here. So there's the equator of the cell. And the chromosomes are now lined up at the equator. So that the best term for it really is equator. And here's a picture of metaphase. Look at the chromosomes lined up, and you can see the spindle fibers here at the poles. Chromosomes lined up at the equator and the poles. You actually have spindle fibers going in all directions, but the ones attached to the chromosomes are the ones that will pull them apart. Here's another picture of metaphase. There were so many pretty ones I couldn't pick one. So here's metaphase. The blue or purplish stuff are the chromosomes. I guess they're blue. And the red stuff are the spindle fibers, and here are the poles. These things are called asters. So there's a pole. Chromosomes at the equator. Cool, huh? Oh, look at metaphase. So here's a section of probably an onion root tip where it's growing a lot. And so you can see the cells in various stages. And this one, the stage that it's in now, is metaphase. You can see this one's just condensing up. That would be in prophase. This one actually is in interphase. You can see the nucleoli, and you cannot see any individual chromosomes. So that would be an interphase, which is not one of mitosis, the mitosis phases. Here it's prophase, late prophase it looks like, and here's metaphase. This looks like another late prophase, early prophase maybe. That looks like interphase because I can't see any chromosomes. So many great pictures I couldn't choose. Ah, that's a cool one. Metaphase, they're in the middle. All right, I think, yeah, we'll finish this up. Anaphase. Centromeres divide into two. And so you have a chromosome here, and you break it, and look, it's being pulled apart. Snap. So the spindle fibers shorten and pull the newly unduplicated chromosomes apart to opposite poles of the cell. So here's a tricky thing. A second ago, that was attached to that, right? So when they were attached, whoops, so here's this one, and here is this one, right? This is what it looked like at metaphase when you had duplicated chromosomes. So this was one duplicated chromosome. That's this, right? As soon as you pull it apart, you can't, this part was a sister chromatid, right? Chromatid. And there's the other chromatid. As soon as you pull them apart, they're not chromatids anymore, they're apart. So this is now one unduplicated chromosome. And that's one unduplicated chromosome. So you can't call them sister chromatids anymore. So the newly unduplicated, because they were duplicated a minute ago, the newly unduplicated chromosomes are pulled to opposite sides. Each pole now has an identical set of chromosomes, and they are all unduplicated. So this is going to be one cell, and that's going to be another, and 1, 2, 3, 4. 4 on this side. If it were you, it would be 46. 4 on that side. And this one is identical to that one, unless you've messed up. That one's identical to that. That one's identical to that, and so on. Here's a real picture of anaphase in a plant cell. Notice you still have just one cell. You don't have it separated out yet, but your DNA is separating. So when it's being pulled apart, that's anaphase. So chromosomes are being separated there. Here's another picture of anaphase. You can see interphase here because you don't see any condensed chromosomes. This looks like prophase because the chromosomes are condensing. I don't see any metaphase here, but that's a really nice anaphase. So quiz time. I think I'm going to pause here because it's getting kind of long.